Elita Jauneikaite, and I'm Assistant Professor in Microbiome and Bacterial Genomics at Imperial College London. Today I will share with you some work I've been doing with my colleagues at Imperial College London, as well as UK Health Security Agency, on Group B streptococcus. Group B streptococcus is a major causative agent in causing infections in neonates globally, and in recent years it has been as well implicated in causing more and more diseases in adults, especially older adults. Primarily, Group B streptococci is actually asymptomatically carried by approximately 30% of people in their gut or urogenital tract. And though associated with neonatal infections, especially in the first week of life, Group B streptococcus as well can cause severe infections in older babies, 7 to 90 days old. There are currently no vaccines available for Group B streptococci, but 2 vaccines are being in clinical trials. One of them is based on the 6 polysaccharides that are on the capsular of the Group B streptococcus, and the other one is based on the 4 proteins that are again on the surface of Group B streptococcus. So with the colleagues, we decided to investigate if over 1,000 whole genome sequences available of Group B streptococcus in the UK between 2009 and 2023, what sort of distribution of serotypes as well as proteins targeted by the vaccine is present in this population. So we did an extensive bioinformatics analysis of over 1,000 whole genome sequences of Group B streptococci and identified that, as expected, majority of the serotypes responsible for invasive disease in various age groups was serotype 3, primarily clonal complex 17, in particularly dominating in early onset disease, which is within the first week of life in neonates, and late onset disease, which is 7 days to 90 days old babies, as well as being one of the dominant serotypes in the adult groups as well. So overall, over 96% of Group B streptococci isolates investigated in the study would be one of the 6 serotypes included in the vaccine, with 97.5% of the serotypes identified in early onset disease and 98.5% of serotypes identified in the late onset disease. Next, we have looked at actually the presence of the 4 proteins targeted by the protein vaccine, the N-terminus of the protein, and have identified that majority of the isolates with more than 90–95% of isolates would actually have at least one of these proteins present on the surface. Again, the distribution of these different proteins was primarily dominated by the Rib protein in the neonatal samples. Rib protein is associated with serotype 3 and clonal complex 17, so that was not too surprising. But as well, we found other proteins present in these data sets, as you can see here. Again, these other proteins are as well primarily associated with the top serotypes identified in the study. Further, we wanted to investigate what are the allelic variants of these proteins, just to see if there's potential implications of that in the protein-targeted vaccine. We have identified that each of the protein had 7 to 16 variants present. However, just to reassure, in each of the protein, 1 variant was the dominant. So all the other diverse that we have identified was present actually in 1, maybe 2 isolates. And to carry on further with the study, we're actually doing a 3D modeling of these allelic variants that we have identified, trying to investigate if potentially they could have some sort of differences in how immune cells are reacting to them. So we have identified that there's a high prevalence of the vaccine targets in our GBS population, which is promising from either of the vaccines that potentially will be used in the future. And as mentioned, we are investigating further to see what potential protein variations are present in this data set as well. So on behalf of myself and my colleagues, I would like to say thank you to all the laboratories in the UK who are sending their GBS isolates to UKHSA, because this allows us to do the work that we're doing. And also I want to say thank you to all the colleagues who are contributing to this project, working together with me.